at her master's thesis project on small mammal microhabitat use and species composition at the first finished wildlife crossing structure on the I-90 project.
Thanks, Chris. Thank you all for coming today. I'm excited to share the past 2 years of my research here at Central with everyone. So, habitat fragmentation involves the division of large continuous swaths of habitat into smaller, more isolated subsets. And this reduces not only the habitat quantity, but also the quality of those remaining patches. So they're more heavily influenced by anthropogenic disturbance. And this can cause wildlife mortality because animals may have a harder time finding food, finding shelter, escaping predation. And connectivity, or the movement of animals between habitat patches, goes hand in hand with habitat fragmentation. So as the landscape becomes more fragmented, connectivity can decrease. And this can further create more isolated populations. So the most extreme case, if populations become extremely isolated, is there can be an accumulation of deleterious mutations and causing inbreeding depression, or simply the genetic makeup of those populations becomes more homogeneous, and then they're more vulnerable to stochastic events or changes. Dispersal, or the movement of animals from their natal patches out to new patches, ultimately is vital for the maintenance of healthy population. And this is especially true with animals with limited mobility or small home ranges. So if an animal already doesn't move very far in its lifetime, having that movement further restricted can have really serious consequences. Transportation corridors, so our transportation corridors like roads and interstates, are physical barriers to animal movement and can reduce connectivity. So they serve as a barrier in terms of if animals decide to cross, they can be struck and killed by a car. Or animals can avoid roads because of all the factors such as lights and pollution and vibrations that will just cause them to not want to come near the road at all. So either way, they serve as a very clear barrier to animal movement. So road ecology is a growing field within ecology, and it is specifically looking at the impact that roads have on ecosystems and populations. So within road ecology, there's been a lot of research done on how roads impact ecosystems, and then also how can we solve those problems in an applied ecology sense. So this can be through the establishment of wildlife corridors, or areas of designated animal movement, protected forest, or on our end, establishing connectivity emphasis areas, or basically designated areas along roads that are deemed high risk for connectivity problems. And these can be determined by looking at previous migration routes or areas where there's a lot of wildlife vehicle collisions. So once we determine what these connectivity emphasis areas are, we can determine the best place to put wildlife crossing structures, which are effective mitigation tools. And these can be expanded culverts, underpasses or expanded bridges, or large-scale wildlife overpasses. And all of these structures have been shown to reduce wildlife vehicle collisions because they allow animals to safely cross the road by either going over or underneath the road. However, the focus of these crossing structures is generally on large mammals because of the large safety risk that large mammals pose when motorists strike them. Therefore, very few studies overall have been conducted on small mammals in crossing structure use, despite them being heavily impacted by the effects of reduced connectivity. So here's a map showing I-90 as it passes through Central Cascade Range. I'm sure we're all familiar with this stretch of road. And I-90 serves as an awesome corridor for our movement east to west. However, it's a really great barrier for animal movement north to south. So I work within the I-90 Snoqualmie Pass East project, which once again, many of you may be familiar with. It's a 24-kilometer, 15 stretch of interstate that is involving lane expansion and other upgrades to reduce traffic congestion and seasonal closures from avalanches and rock slides, and ultimately to improve the safety of this stretch of road. Additionally to the lane expansion, they're also implementing and upgrading more than 30 wildlife crossing structures to address the effects of reduced connectivity on wildlife. As part of this, they've also implemented pre- and post-construction wildlife monitoring to address if the mitigation effects are actually effective for wildlife. And not only are they looking at large mammals, but also small, low mobility species such as small mammals, amphibians, fish, and reptiles. So here's a picture showing all of the crossing structures, or some of the crossing structures along the I-90 project. My study focuses at the Gold Creek Connectivity Emphasis Area, which is at the western tip of the project area. And in this region, they've expanded both the Gold Creek bridges and built the Hayek Wildlife Undercrossing. So this is an aerial photo showing the Gold Creek as it enters into Lake Ketchelis. And Gold Creek was deemed an area of high connectivity problems because the valley basically acts as a funnel pushing animals into I-90 and Lake Ketchelis. So not only did they expand the bridges here to improve connectivity, but also built the Hayek Wildlife Undercrossing here. So here's a picture on the ground of what the undercrossing looks like. So it's an entirely terrestrial crossing structure. It is about 40 meters wide, 60 meters long, and 55 meters tall. It has a variety of habitat features within it. So there are rock piles here that line both the eastern and western borders. There is native plants, soil, and a kind of a barky substrate on the ground, and then also lots of fallen logs. And these were all implemented to encourage animal use. So within road ecology, there's been a lot of research done on what makes a crossing structure effective for a variety of fauna. And what we found is there's a lot of species-specific factors that influence structure effectiveness. Some of these may include the dimensions of the structure. So for example, grizzly bears, wolves, elk, and deer prefer structures that are high, wide, and short, whereas black bears and cougars tend to prefer structures that are more narrow, like this expanded culvert here. Additionally, the presence of natural vegetation has proved to be very important to increase animal use of crossing structures. And this involves not only the distance from the forest to the crossing structure, having lots of vegetation and cover, but also vegetation within the crossing structure improves use. And this study was conducted on small mammals specifically. However, this study, again in 2016, was a small mammal study looking at martens and weasels, and they seem to prefer crossing structures that are slightly more open, which makes sense if you think about the natural history characteristics of the different species here. Additionally, the presence of a natural substrate is important, so instead of having a concrete bottom or metal culvert bottom, having soil or a woody bark or vegetation all influences crossing structure use. And then lastly, addressing the natural history and what habitat features the species that you want your crossing structure to be used by. So once again, this is my site. This is one of the restoration sites. And you can see that these restoration site and the crossing structure have many of those features that I just talked about. So presence of a natural substrate, vegetation, presence of habitat features such as rock piles, fallen logs, brush piles, and snags were all constructed by Washington DOT to increase the effectiveness of these crossing structures. So if it's been determined that the most vulnerable species to reduce connectivity and road mortality are those with larger body size, high mobility, and lower reproductive rates, why should we study low mobility species? Well, for small mammals in particular, they play a really key role in ecosystems. So they're centrally located within the food web, which makes them not only prey species, but also dispersers of seeds and fungal spores. They burrow and dig through the soil, which turns it over. Additionally, since they have shorter generation times, they respond very quickly to change that we can observe as ecologists. And typically, when we track them, we can get really high population sample sizes because they occur in higher population densities, which increases our power to draw stronger conclusions. So small mammals are very generally considered to be less than 5 kilograms. Some advantage to being a small mammal is they can eat an abundance of food. So there's many different foraging potentials. They can utilize many different microhabitats that their larger counterparts cannot, simply due to their smaller size. They can also escape, so their strategy is to hide rather than flee. And they have generally higher fertility. However, some disadvantages to being smaller is having a shorter lifespan. There's a much higher energy cost due to having a higher surface area to volume ratio, and limited mobility, limited dispersal. Some of the small mammals within my study are insectivores, such as shrews, lagomorphs, which would be pikas, rodents, and hares. And then the largest group would be rodents, which are chipmunks, mice, voles, flying squirrels, and Douglas squirrels. So within the small mammal group, there's lots of habitat specialization. So we have some habitat generalists, which are generally considered to be very tolerant of their environment, have very flexible environmental requirements. Some of the species in my study that would be characterized as generalists are deer mice, long-tailed voles, and the montane shrew. These are all able to live in more open environments. Whereas we also have habitat specialists, which are species that have more specific requirements of their environments. And these are all closed canopy or mature forest specialists. And some of these include Douglas squirrels, redback voles, chipmunks, of course the charismatic flying squirrel, and the cinereous shrew. So this brings me to my objectives, which was ultimately to compare species composition and richness between the crossing structure, the adjacent restoration sites, and then compare that to the nearby forest, with the forest being the baseline small mammal composition I would expect. Additionally, I was curious to assess how small mammals were using habitat features in the crossing structure and the restoration sites, and compare how those constructed habitat features were potentially used differently than the features in the forest. So I predicted that the undercrossing would have higher relative abundance of generalist species because of the higher level of disturbance that's in the crossing structure since it's so close to I-90, and would have lower species richness compared to the adjacent forest sites. I additionally expected that the restoration sites would be relatively intermediate in species richness between the crossing structure and the forest because the restoration sites are a little bit farther away from I-90 and a little bit closer to the forest. Lastly, I expected that we would capture small mammals when traps were closer to habitat features than traps placed in more open areas. So specifically, my study area, once again, is on the I-90 corridor, and specifically, my sites are at this Hayak dot here. So if you zoom in, I have the Hayak Wildlife Undercrossing here that's directly underneath I-90, 2 adjacent restoration sites, one north and one south, and 2 forest sites, one north and one south. So starting in July 2017, I employed 3 methods in order to get the most comprehensive assessment of small mammals at my sites. So I used not only live trapping, but also installed wildlife cameras and track tubes. So for live trapping, we trapped each grid 2 times during the season, so once in the beginning of the season and once at the end of the season. And each grid was trapped for 2 consecutive nights. We used tomahawk traps, which are these larger metal cages, Sherman traps, which are slightly smaller, and then installed pitfall arrays here, which is a bucket in the ground. This came to a total of a little over 1,300 trap nights, which is a measure of how many traps we have multiplied by how many nights traps are open. And we took, for every animal capture, we took a variety of measurements and collected demographic data for every species. Additionally, we took a genetic sample and pit tagged every individual, which was really important because this allowed me the opportunity to differentiate individuals. So this is what a trapping grid looks like. So it's a 7x7 grid that's 60 meters by 60 meters. So there are Sherman traps placed 10 meters apart, tomahawk traps placed 20 meters apart, and then 2 pitfall arrays, one in a more upland area and one in a lowland area. Now, if we place this grid in the forest, let's say, or at one of the restoration sites, here's what the matrix of habitat features may look like. So you can see at each trapping station, there are potentially a variety of habitat features. And what we did is we walked around, and at each trapping station, we determined what habitat feature was closest to it. So for instance, this A1 would be a rock pile, A6 here would be closer to a brush pile, E5 would be a fallen log, and then all of these other ones would just be open. So this is a picture of what these look like in real life in my actual grids. So here at a restoration site, this would be qualify as an open area. So it's important to mention that an open area is not that the canopy is open, it's more so that there isn't the presence of those other habitat feature types. So for example, here's what an open area looks like in the forest. So there's still canopy, there's still some vegetation, but there's just not a clear habitat feature type. Here's a picture of rock piles at my restoration sites, brush pile in the forest, Brush piles occurred at all sites, whereas rock piles were only at the restoration and crossing structure sites. And then fallen logs here within the crossing structure, and then here's a few in the forest. So in addition to live trapping, we also installed track tubes. These were placed concurrently while we were live trapping, and there was 12 to 18 tubes per site. And the way we divided them up is tubes were only placed at stations that had habitat features, and those habitat features were, or those stations were randomly selected. So for 3 randomly selected stations, we then placed 2 track tubes to try to get as many tubes around each feature as possible. And then tubes were placed depending on what habitat features were available at what sites. So here's an example of what a track plate looks like when it's done. So this is a Douglas squirrel print. We had a curious juvenile. This is the only one we captured. And then more typically, I had lots of vole tracks here. Our last method that we used were the installation of wildlife cameras. These were placed for 2 weeks at each grid, and these were placed when we were not actively live trapping at our grids to avoid any sort of influence by researcher presence. And we installed 8 to 16 cameras per site, and this was entirely dependent on what habitat features were available. And we also put some cameras at snags to kind of see if small mammals were using snags. This was our only way to test that. So here is a Douglas squirrel at a rock pile. The only animal we caught on a snag, which was a deer mouse running up and down a snag at night, and a chipmunk here in a brush pile. So for my analysis, for looking at species composition, I combined my 3 site types, so undercrossing, forest, and restoration, and then combined all methods, so that way I could capture as many species, or kind of combine all of those methods together. And unfortunately, I couldn't do any sort of particular statistical analysis because I was looking at species richness, which is just a count, and comparing that between 3 site types. But I did attempt to standardize my captures when comparing differences between the types by either looking at captures per 100 trap nights or proportional abundance. For looking at habitat preference, I ended up only using live trapping data, and I combined crossing structure with my restoration sites to then compare constructed habitat features to natural habitat. I used ultimately a generalized linear mixed model because I had fixed and random effects, and I used a zero inflated model. Because I was looking at count data, it also followed a Poisson distribution, but I had lots and lots of zeros or no captures, so a zero inflated model was more appropriate. So ultimately, my model was captures, number of captures at each station as a function of site type, what habitat feature, and trap type. And then I used the random effects of grid. And then lastly, I did a chi-square post-hoc analysis. Chi-squared is the standard post-hoc for generalized linear model with Poisson distributions. So in total, we captured 469 total animals. 307 of those were individuals, and that came to about a 35% recapture rate. Across all grids, we captured 10 species through live trapping. With wildlife cameras, we caught a little over 800 images with wildlife. I then narrowed that down based on standard protocol to 350 visitations. And a visitation is defined as a photo of a species less than or greater than 1 hour apart of a photo of another species. So, for example, if I caught a chipmunk at 8 o'clock, I couldn't count an image of a chipmunk as a second capture until after 9 o'clock. Additionally, we used track tubes. I had about a 75% success rate, so pretty good occupancy of those, with 116 visitations. For track tubes, a visitation is number of species in a tube. So this is a mosaic box plot showing proportional abundance of the species we captured between my 3 site types, so forest restoration and WCS is wildlife crossing structure. So right off the bat, you can see the forest captured the most species, so we had a species richness of 11. The restoration site was a little bit less, with 8 species captured, and the crossing structure, we only captured 4 species, all of which were generalist species. So next I'm going to look at some species-specific trends. So the long-tailed vole, an open grassland, open canopy generalist, increased by about 14-fold from the forest to the crossing structure. The maniculatus deer mouse species increased by about 5-fold from the forest to the crossing structure, another open canopy generalist It's a little more of a combination. It can live in the forest as well. The montane shrew, once again another generalist, increased by 200% from the forest to the crossing structure. And then lastly, the kenai species of deer mouse increased by 38%, but roughly these proportions were equivalent between the 3 site types. Since we pit tagged, we were able to observe movement. So we found 6 movement events, all were deer mice species. So our largest movement was 136 meters by an impressive female from the northern restoration site into the crossing structure. The remaining movements were from the crossing structure out into the southern restoration site. So looking at comparing my method types, I kind of want to briefly talk about how all 3 methods captured deer mice in highest abundance, with live trapping capturing significantly more. It appeared that live trapping seemed to underestimate the number of bulls compared to my other methods, but then captured more shrews than the other methods. And what was exciting, and the kind of the purpose of employing all these methods, was capturing species that we couldn't get with any other method. And that was true with the snowshoe hare, which was only captured on camera and was captured at 2 grids. Additionally, we captured Douglas squirrels and flying squirrels and chipmunks in higher proportional abundance with cameras than any other method. So these are histograms of number of animals captured. So we had 0 would be no captures at a trapping station, and that went up to 4 captures per trapping station, and this is the frequency or how many times those captures occurred. And then we have naturally constructed habitat features versus artificially constructed habitat features. And then we have brush piles, fallen logs in natural compared to we add rock piles into our constructed habitat features by Washington DOT. So my model did not find a significant difference in habitat feature type. So there's no small mammal. Small mammals are not more likely to be captured in brush piles than fallen logs or rock piles. And there was no difference between naturally versus constructed habitat features. But what was interesting is that in these open sites, there was really high frequency of no captures. So this kind of led me to think about, well, what if I combine the presence of a habitat feature, so fallen logs, rock piles, brush piles, into one category, and compare that to these open features. So this is the same style of histogram, and I did find that small mammals are more likely to be captured when a habitat feature is present than when it is absent. And once again, you can see lots of no captures in these absent piles, whereas in present, it's more equally distributed. So we found the lowest species richness in the crossing structure. And this is likely due to the higher degree of anthropogenic disturbance of the crossing structure itself. So it sits directly underneath I-90. So there is vibration, noise, pollution, lights that are all making it slightly harder, more challenging for many small mammal species to occupy this habitat. Additionally, this crossing structure is only 2 years post-construction. So we could define it as kind of an early successional environment. So it could just be that animals need a little bit more time to move into this habitat. Additionally, we found increased abundance in generalist species the closer we got to I-90, so with the crossing structure having higher abundance of generalist species. And this could be because the restoration sites can be qualified as a more open canopy habitat, which would favor our open canopy generalist species. And additionally, since it's in that early successional environment, 2 years post-construction or post-disturbance, it has been found in other studies that our species, Paniculatus and Longicatus, really tend to thrive and increase in abundance in these early successional post-disturbance environments. Additionally, I want to briefly talk about the effect of edge effects, which is the increase of abundance of small mammal species that we can observe on the edges of disturbed habitats. And animals, we may observe some species that having an increase of abundance when species' main predators tend to avoid roads. So it may be beneficial for a species to occupy this more degraded, disturbed habitat if they're going to be less predated on. Or since often we plant grasses or other sort of food along roads and interstates, there can be abundant food. So once again, if you can occupy this more disturbed habitat, it's advantageous for you to move in there. However, some species, like maybe some habitat specialists, we're going to see a decrease in abundance because they're just going to avoid the roadway because it's too much of a barrier to occupy that habitat. This was a study done in 2015 that looked at small mammal trends in general and then also looking at species-specific trends. So once again, they looked at, very similar to my study design, the abundance of small mammals within a crossing structure, within the entrance, and then in the forest. And they found that in general, all small mammals kind of had the same proportional abundance or the same presence no matter which site. But then when they divvied up and looked at the species-specific trends, they found that deer mice, our generalist species, were highest in the crossing structure and then decreased from the crossing structure to the forest. Redback voles, which are mature forest specialists, were lowest in the crossing structure and increased dramatically into the forest. And then meadow voles, which would be similar to the long-tailed vole in my study, was highest at the crossing structure entrance, which would be a more open area. My second objective was addressing habitat feature use. So we didn't observe any specific feature preference. So small mammals were not more likely to be captured at any specific feature. So once again, fallen logs, brush piles, rock piles. And this may be due to the higher abundance of generalist species that we captured. So since generalist species are really flexible with their environmental requirements, tolerant of a variety of conditions, they may just not really care which habitat feature they're using. But some studies have found that small mammals and generalist species tend to prefer fallen logs and branches. So it could be that the presence of a habitat feature is just preferred over nothing at this point in time, since that habitat is still needing some time to mature and develop. And these open areas may just be entirely too risky for animal movement at this time. So moving along any feature, occupying any feature is going to be better than occupying or moving through those open areas. And potentially, as the vegetation matures in the restoration sites, this could change. So this was a study in 2004 that was specifically looking at what characteristics are most effective for small mammal crossing structure use. And this was a translocation survey. So they took animals from one side of one side of the interstate and moved them to the other side, and then looked at the characteristics of crossing structure and if they returned home. And they found that overall, as the cover increased, the proportion of small mammals that returned generally increased. And this was especially true for the specialist redback vole species that really depended on having that heavy cover to use the crossing structure. So briefly, I want to evaluate the different techniques that I used. So for live trapping, it was really advantageous for us to be able to identify individuals. It allowed us to make stronger conclusions about what the proportional abundance was of the populations we surveyed. And uniquely, we were able to track movement. So we were able to see movement between grids that we couldn't have otherwise. However, it is very time-consuming and relatively invasive on the environment because we are specifically handling these animals and stressful for them. With track tubes, we were able to identify some animals or some tracks to species. The materials are very cheap. It's just pieces of gutter and duct tape. And they're very easy to install, basically just place them on the ground. And they are not a non-invasive technique because we don't have to handle animals to get tracks. However, sometimes the IDs were really challenging with the track tubes. So I don't know whether it was one mouse that was very excited or maybe there were 50, but I would have track plates that were just covered in tracks. So it was very challenging to figure out what to kind of do with these track plates that were basically totally covered. My last method was wildlife cameras. So this has very, very high capture success with minimal effort. So I would come back with hundreds of photos of small mammals that I could then do a lot of different things with. And it increased the likelihood of rare or hard to catch animals, which was specifically why I wanted to employ them. So for example, we captured snowshoe hare on camera that we didn't capture with any other method. However, at this point in time, cameras are still very expensive, and it can be challenging to differentiate individuals and differentiate species. So in summary, small mammals are moving through and living in the crossing structure. At this point in time, we only captured 4 species, all of which were generalist species. And as the vegetation matures and this environment becomes a little more complex, this number should change. Small mammals are using habitat features, although we didn't observe any specific preference for one. And these habitat features provide critical habitat and encourage safe movement through the crossing structure, which is the goal for us trying to improve connectivity for these small mammal populations. So features should continue to be included in future crossing structures, as shown by my study and also a huge body of literature. Lastly, my take-home point is, unlike their large mammal counterparts, small mammals are not only moving through, but also occupying the habitat that we're creating for them along these roadsides or within crossing structures. So the quality of habitat is really important for the maintenance of healthy ecosystems. I'd like to thank my phenomenal advisor for taking me on and leading me into the small mammal world, my committee, Dr. Beck and Dr. James, my awesome field technicians, JL and Jordan, and the other volunteers who helped who are in this room, my graduate cohort for constant advice and support, and then Washington DOT for funding my project. They were the main supporter financially, the U.S. Forest Service, and Kittitas Audubon Research Grant for also providing financial support, and the biology department for providing facilities and their phenomenal staff. Any questions?